Don Coffey was really a legend for urologic research in this country. He was, from 1974 to 2004, was the director of research at the Brady Urologic Institute, and his scientific accomplishments were many, but he's mostly known for mentoring truly a generation of leaders in cancer research, and he would be famous for questioning them with his advice of, if this is true, what does it imply? One of his foremost disciples was Alan Parton, who we lost this year after a battle of his own cancer. And he served from 2004 to 2021 as the director of the Brady Urologic Institute. And similarly, while his scientific accomplishments were many, what he is mostly known for is mentoring a generation of urologic surgeon scientists. So this is both an ode to these two sort of giants in the field, and sort of a question of what's to come within our own field. So to that end, we are both the mentors and we are the mentees. And so we're gonna take three topics related to, but not necessarily about bladder cancer. So what do these advances mean for our field and our patients? What comes next? So our topics for today are going to be drug delivery, epigenetics, and mRNA vaccines. And I would ask that this group be engaged and feel free to come up and ask lots of questions, and with that mentality of what's going to be coming next. So our first speaker is Dr. Laura Ensign, who I've had the pleasure of knowing for many years, and who has, I think, one of the most rapid promotions to full professor in Johns Hopkins history, and is widely known for her work in drug delivery in many different organs, in the GI tract, in the vagina, in the bladder, in the eye, and we're really excited to hear what she has to say. Next, we have Josh Meeks, who does not need an introduction to this group, but one of the things that I personally really admire about Josh's work is that it really hits the breadth of bladder cancer inquiry. So whether it's subtypes, or in this case epigenetics, there's so much to learn from Josh, and we're really excited. And last, but certainly not least, Vinod Balachandran, who's an attending surgeon at Memorial Sloan Kettering and a true surgeon scientist. I'm thrilled that we have two surgeon scientists up here speaking today as people we can all look up to. And what he's widely known for, of course, is mRNA vaccines in pancreatic cancer. And so between these three, I think we're going to have a very rich discussion and learn a lot. So without further ado, I'll have Dr. Ensign come up.
Okay, thank you for the invitation. So as Max mentioned, I'm actually a professor of ophthalmology, but I am an engineer by training, so I have a PhD in chemical and biomolecular engineering. So I'm the vice chair for research at the Wilmer Eye Institute, and largely my work works on ocular drug delivery, but as I'll talk through, we work on many different diseases and organs because drug delivery is really something that it can be applied to just about any part of the body. And so this is also a great meeting because the work, drug delivery by nature, it's very interdisciplinary. We have to collaborate. And the work that we do, the only way that we can have impact is by, doing needs identification and kind of dreaming with physicians on what's the next things that we can do for patients. So I'll tell you a bit about a few things, kind of breezing over a few things that we've been working on in the eye, and then even one case where we did translate that over to the bladder pretty successfully. So just financial disclosures about some companies, two of the technologies, those are being developed commercially. So the whole point of drug delivery is just this concept that, everywhere in our body we have barriers, which is a good thing, right? It's the way that we stay healthy, we keep viruses out, bacteria out. And so these barriers then also make therapeutic delivery and treating diseases very challenging. And so for the eye, it's, especially a very important part of our body, right? We don't want to have foreign material entering our eye. So whether you're talking about topical delivery or delivery from the systemic route or any kind of injection, there's many, many ways in which our ocular tissues are designed to keep drugs out. And so a lot of what we do is then just taking drugs that have known action, that are used for known purposes, but we're making them work more effectively. And so why this is necessary in most diseases, but particularly in the eye, is because of non-adherence, right? So in ocular treatment, that's a particularly problematic issue where patients, especially with eye drops, may not adhere to the treatment regimen. So specifically within ocular diseases, non-adherence estimates range from 30 to 80%, and then in certain cases, non-adherence then leads to actual progression of disease and blindness. And particularly with eye drops, I'll mention a couple different eye drop cases. The more often a patient has to use eye drops, the more likely they are to not adhere to this regimen. And then with injections, also there's been irreversible vision loss shown when patients miss their injections. And so the group that I work in at Johns Hopkins is actually part of a center. So it's a very unique entity, right? Because we're largely a bunch of engineers that are in a department of ophthalmology. We all work on drug delivery, and so we have a whole center for nanomedicine that does drug delivery within the department of ophthalmology. It's kind of unique. It's certainly unique within ophthalmology, but I think also just in clinical departments in general, it's a very unique group. And so we work on all kinds of different drug delivery systems. Most of them are nanoparticle based or have some sort of nano dimension. And as you can see, we actually don't just entirely work on, and I don't know if this probably won't show up there. So my group does a lot of work in women's health, also in gastrointestinal diseases, and basically anything that has a mucosal surface or where you can inject into tissue. So you can imagine that that's pretty far reaching. One of the important aspects of what we do also is to be heavily translational. As I mentioned, we're always doing needs identification and trying to design our drug delivery system so that we can actually do product development and eventually get to patients. And so there's just a cluster of startups in the lower corner there where we've been patenting what we're doing and then starting companies to develop into clinical trials. And so for the eye, for those that aren't familiar in the audience, there's many different routes of administration, topical being the most common, of course, eye drops, and probably about 90% of commercial formulations are eye drops. So these have obvious advantages. They're relatively easy to use. The patient can do it themselves, but then we have to usually prescribe drops to be dosed very frequently because the clearance and ocular bioavailability is very poor through that approach. There's lots of different routes of injection, subconjunctival, which is kind of around the white part of the eye, intravitreal, going right into the globe of the eye. But of course, as you get more and more invasive with these injections, then you have more likelihood of infections and other kinds of negative side effects. And so there's also implants, and this is where also sustained release has started to really come out. Like Ozurdex has been out for a while, but there's also Durissa and some other implants that are coming out and getting FDA approval. But of course, more surgical insertions usually, and then the excipients, the carrier, that implant material itself, and the degradation can be an issue, buildup of products. And so I'll also just mention, because this is related to the first story I'm going to tell you about ocular melanin, so the pigment that's in our eye. So our eyes are actually full of melanin. I mean, you're probably more familiar with the iris. The pigmentation in your iris is also due to the amount of melanin that you have. But within the eye, there's a tremendous amount of melanin to absorb light and radiation that's coming through, right, to protect our retinas. And so interestingly, if any of you have ever had children that were born with blue eyes and then their eyes turned brown by the time they were 2 or 3, it's because the melanin was continually being produced after birth. And then your melanin production stops after that point, and so all of the melanin in your eye you'll essentially have by the age of 3, and it does not, relatively little turnover. And so why this is interesting then is that this can act as a sustained release depot, if you will, if you have drugs that can bind to the melanin pigment that's in your ocular cells. And so I'll talk a bit about how we utilize that. So this was a recent story that was actually published in the past few months, but we came up with this concept of trying to use machine learning to design peptide sequences so that they could bind to particular structures within the eye. In this case, we were designing peptides that could bind to melanin. And so then, what this is basically showing is we came up with a fluidic microarray to generate the binding data, right? With anything that's machine learning, you need a training data set. So we were generating, how does peptide sequence relate to melanin binding? And then using that to train a machine learning model that could then tell us, okay, well, this sequence would be predicted to bind very strongly. And we could also then have the machine learning algorithm tell us, well, what would also be cell penetrating, what would also be non-toxic. So we developed these multifunctional peptides that we could then attach to small molecule drugs to change their pharmacokinetics in the eye. And so what this is just showing is basically then the predicted binding to melanin versus the experimental binding to melanin, just showing that there was good correlation there. And then this is then moving into cells. So melanin is contained within organelles inside the cell. So you also have to first get inside the cell to then reach the melanin. So on the left, it's showing if the peptides were not cell penetrating, and then on the right or in the middle is if they are cell penetrating. Just showing that we could get increased binding based on the predicted binding if they could both enter the cell and then bind to the melanin. So by having this dual feature of cell penetration and melanin binding, we could get very high accumulation of peptide within pigmented cells. And so then moving into rabbits, so with a drug brimonidine, so this drug is used for intraocular pressure lowering. Typically it's dosed 3 times a day as an eye drop. So the triangles that are compressed into the corner of this intraocular pressure curve, that's a single eye drop that works for about 6 hours maybe before the IOP goes back to baseline. And then we demonstrated with this peptide conjugated to the brimonidine, when we do an intracameral injection into the front of the eye, we can get it to last for a couple weeks. So we're working now on how to extend this out to several months. And so you can imagine then this strategy could be applicable to basically anything where you could develop an in vitro screening system to look at binding events, right? So as long as you have enough peptides and you have, some sort of target in mind that you're trying to bind to, then you can do similar kinds of approaches to do this multifunctional engineering. And so then to get into eye drops then, the question is why do we have to use them so often? So of course we have reflexive blinking and then tear production. And every time I talk about this slide, I start reflexively tearing just thinking about it. And so our eyes are very good at cleaning the surface, right? So as soon as you put an eye drop in, you're gonna be blinking and tearing to clear it. And so there's lots of different strategies that are used pharmaceutically for improving topical ocular drug delivery. So there might be viscosity enhancement. If it's thicker, it'll stay there longer. The drug will be absorbed for longer. There's penetration enhancement. So as this you can imagine this sounds like if you're opening up tight junctions and forcing drug through, that can be toxic over time. So that's one of the drawbacks of this approach. And then solubility enhancement. So for drugs that are very low solubility, if we just get them into solution, then you have a bigger gradient and you can get more absorption that way. So we have a drop vehicle that we're working with that actually kind of combines all 3 of these features, but without being toxic. And so the way that this works is it's a thermoreversible polymer. So it's a solution at room temperature, and then it forms a gel when it gets heated to body temperature. But the way that we're formulating it actually is so that this polymer is in lower concentration than what would typically gel, and then also hypotonic. So water gets absorbed by the ocular surface. And so as the water is being absorbed, then you have concentration of the polymer, and then it forms a clear gel that's just kind of uniformly spread over the ocular surface. And so whatever drug that it's carrying with it, it has this thin film that will stay on the ocular surface. And so this just kind of shows it on the surface of some rat eyes. So in that top row, this is kind of like a conventional thermogel, gels right away. The red line is outlining the gel layer on the surface of the blue cornea. And so you can see that's very irregular, right? So you can imagine that as soon as it contacts the ocular surface, it gels immediately in place. And then our eyelids just wipe that away and it gets caught in our eyelashes and things like that. On the bottom, you just have like a viscous liquid eye drop. So you can see it on the ocular surface immediately, uniformly coating the surface, but then when you blink, it disappears, right? It just gets wiped right off. But our approach then, it'll form that in the middle, it'll form that thin, uniform, clear layer, and then it's resistant to blinking though, because your eyelid just passes right over it. And so what we've then shown is that, we can improve the delivery of water-soluble drugs, and you don't need to pay too much attention to the details because this will get busy, but just that we can do better than water-soluble eye drops that are on the market, alpha-GAN-P, without increasing the systemic absorption of drug. We can solubilize very hydrophobic drugs, so ones that are commercially available as a milky suspension, we can make it same drug concentration but clear, and then also last for longer. And then also even improve delivery to the posterior segment, so the retina and the tissues in the back of the eye, which is really difficult to do with an eye drop. And then also just, again, being non-toxic even with very repeated dosing for up to weeks and months. And so this technology is actually being developed by our startup now. And it has lots of different applications in other mucosal surfaces as well. So I'm just going to finish up with an example then that we did actually apply to the bladder with Max when we were years ago, I guess, when you were a research fellow. So this technology it's called the mucus penetrating particle technology. Kala Pharmaceuticals actually has a couple approved eye drop products based off of this. And so we've used it to deliver drugs to the respiratory, gastrointestinal, reproductive, and urinary tracts. It's based on the concept that mucus on mucosal surfaces is a barrier, and that it can either be a steric barrier because it's like a web, or it can be a chemically adhesive barrier. So for particles, like a conventional nanoparticle, it's going to be a polymer that's going to be sticky to mucus, and so if you apply it onto a mucosal surface, it will be trapped and cleared really quickly. But the mucus penetrating particle technology, we coat it with a non-adhesive polymer, and so what that allows then is that the particles are able to navigate their way through the mucus barrier and then more effectively deliver drug to the epithelial surface. And so this is an example of the mouse vagina tissue. So these are cross-sections showing particles that are stuck in the mucus, the conventional ones, or the mucus penetrating particles are able to coat the epithelial surface and release drug there. This is the airways, same idea. And then this is in the colon of mice that have inflammatory bowel disease, where you can actually see uptake of particles into the broken epithelial surface. And then this was the example with the eye. These are administered as eye drops. They've increased residence time and intraocular drug absorption. So that's what Kala developed into a couple eye drop products. So we found then, as we've been looking at this at different mucosal surfaces, that that surface coating matters a lot. How dense the polymer on the surface is. Essentially, can you resist those mucus interactions? And so then also the tonicity of the vehicle that we're dosing. I mentioned water absorption, right? That water absorption effect is really important and helpful in getting particle delivery on the epithelium as well. But when we encountered the bladder mucosa then, we thought that we knew a lot about how to predict how we could deliver drugs effectively, but the bladder mucosa is very specialized, right? And some of you may be more familiar with this than I am, but there's a glycocalyx but no soluble mucus layer, so it's not like the eye, it's not like the reproductive tract. And so actually then the tissue is very impermeable, right? We can't have waste being leached back out into the body. And then also the bladder undergoes tremendous amounts of stretch and then voiding, and can deal with wide ranges of pH and osmolality. So it doesn't respond to different tonicities the same way that any other epithelial surface does. And this tremendous mechanical stretch and the way in which the membrane is actually expanding and then contracting is really unique as well. So what we ended up finding out as we were stumbling our way through this is that we could utilize these special properties to actually get our particles to be taken up into the tissue and go submucosally. And so that's really advantageous, especially in the context of bladder cancer, right, in terms of getting tissue penetration. So if we manipulated both the particle and the vehicle, we could get this really nice particle uptake and superior drug administration and absorption. And so then in this paper, where Max is the first author, right, that we showed that not only could a nanoparticle of cisplatin, so cisplatin being something that's very toxic locally, and that's shown as CDDP here, if you put it into a nanoparticle, it actually reduces the toxicity of the drug. So these are healthy mice where we are just repeatedly either administering free cisplatin or particles, and you can see based on bladder weight, so there was this tremendous edema and fibrosis that was occurring in response to the drug. But if it was contained within a nanoparticle, then it had minimal effect on the healthy tissue. And then when it was dosed to rats that had chemically induced cancer, that it was able to more effectively prevent kind of the development to invasive cancer in this model compared to the free drug. And what's not shown here is it also reduced the systemic exposure of cisplatin, so safety from locally and systemically. And so then in conclusion, I just want to re-highlight that effective treatment of human diseases is limited by biological barriers and also patient adherence, and this is what really motivates us in the drug delivery field. So nanomedicine sustained release formulations can circumvent these issues that we have with our conventional dosage forms and make drugs more effective. So what we often see is that this biological redundancy that we have can be leveraged to translate different delivery technologies across all different diseases affecting very distant parts of the body, and that for all of this above, that really the interdisciplinary teams are key for the needs identification and designing innovative solutions to these unmet needs. And so just some acknowledgments. So as our group here and a bunch of the students, so we have a lot of grad students and fellows that do all of the work, and I'm very grateful to our funding sources also as well. And thanks to Max very much for the introduction and for the collaboration, and I welcome any questions.
I'll get us started. In bladder cancer in general, the most common treatment for patients with bladder cancer is an intravesical therapy. Okay, it's BCG, although now we're giving several other drugs intravesically. And when we get up at meetings and we talk about new therapies for bladder cancer, we tend to focus on on sort of the biology and the drug targets and the activity, and I don't think we necessarily focus a lot on the delivery into the bladder, which is the most common way we give. We make assumptions about systemic agents, and I'm just wondering, what your comments on that? What other organs? Maybe it's the eye, maybe it's others that have that similar sort of scenario, and any lessons learned from that?
Yeah, those are really great points. And so this is something that we always talk about kind of in drug delivery, is that your drug can be great and your drug can act really well on its intended target, but if you're not getting it to that target and then getting enough of it there where you're not having dose-limiting toxicities, then you can never actually really know whether that drug was effective or not. And so a lot of clinical trials, we believe, fail, for those reasons, right? And so it's actually even something that, even preclinically, we'll develop nanoparticle and delivery systems just to probe biology and look at epigenetics and look at things like that, like the actual effects of a drug in vivo, in a way that's more closer to being able to just bathe a cell in vitro in a dish. And so I think that's super important. There's been lots of other cases, like a lot of the work I do in women's health is very analogous, where so often it's just a question of like, well, if you didn't get the drug there and it can't have its action, and then you can't see the intended effect. And so I think it's a problem kind of in really any disease where we do a lot of systemic and oral administration, right? Because these are the most common approaches that we have. But then, so the way that biological redundancy can also very much work against you is when you have target engagement other than where you wanted it to happen, right? And that's the most common issue with immunotherapies and chemo, right? So I feel like I could lecture on this point for the next hour, but I'll just kind of end it there, that it's a really, really useful point. And delivery is really, I feel like, the step in between drug discovery and then getting to actually like effective treatments.
So you mentioned that the normal urothelium has a glycocalyx but not mucus. Do you know much about like urothelial tumors and whether they possess the same properties? Because that's really what we wanted to target ultimately, at least at this think tank.
Yeah, that's a really good question. And so there's certainly plenty of literature that suggests that the glycocalyx and mucins play a big role in a lot of cancers, including urothelial cancers, and that there might be overexpression of certain mucins in the glycocalyx and things like that as well. So I would say that actually in, thinking about mucosal delivery and how to overcome the epithelial barriers, it's even more of an imperative in the setting of a tumor. But then also if you've scraped out the tumor, right, and then you're dosing chemo after that, really getting tissue penetration where you've opened up the submucosa is really important. And but that's also, again, it's where the particles can really start to show a benefit, because those intertissue barriers are even a lot of times more formidable than the secreted mucus layers.
Thank you.
That was really neat, thanks. Phil Bosch from Philadelphia. Two, I think, quick questions. One, how long do these substances linger after you put them in? And second, and you don't have to, reveal any trade secrets, but is this something you anticipate conjugating to existing drugs, or making new drugs that you put with your polymers or whatever they are?
Those are both really good questions. So in the context of dwell time, it very much varies depending on what the carrier is. So in settings where we might be making, like, the particle might be entirely drug with just a little bit of polymer coating, in which case all of that will clear out very rapidly. In some cases where you're talking about actually like biodegradable polymer shells, then you have to wait for the polymer to be broken down enzymatically. We're definitely way away from any kind of inorganic materials, like where those can hang out and they can accumulate in the liver and things for a really extended period, like gold and all those other kinds of materials. So we're on the biodegradable side of things. And then your other question, I think, was about kind of what we can deliver, right? And so a lot of times we'll start with drugs that are known, and one that we just want to improve the delivery. But then also with existing systems that we know have some interesting benefits, we might pair those with like new drugs where we're actually just trying to look at mechanism and engagement and show proof of principle of the drug itself. Sometimes we're even doing siRNA or mRNA delivery just to look at mechanism. So the delivery is really just like, how can you look at the biology in a more effective way?
Thanks.
Beautiful work. Of course, I'm familiar with it. Just a comment is that kind of, I guess, through preclinical trial and error and other approaches, the whole development of Adstiladrin really depended on detergent Syn3 and its ability to penetrate the layer you've been targeting. And I think a lot more work should be done on this area to help exploit the fact that we can do local therapy in urothelial cancer.
Absolutely, absolutely. Yeah, that's great. Thank you.
I'm going to just ask one more question because we have the time, and that you brought up something that I wasn't necessarily thinking about, which is the use of drug delivery to reduce toxicities in addition to improving efficacy. Is there, I guess, any specific cases from either other organ systems where you've seen that work?
Yeah, for sure. And one example is actually related to one of our startup companies. So we were doing intravitreal, so intraocular injection of a sustained delivery system. It's a microparticle that has a small molecule, and it's slowly eluting it over time. But it was a cancer drug. It's sunitinib, right? And which prevents vessel growth. And so in AMD, you have aberrant vessel growth. And so you would never give, systemic sunitinib to someone with AMD to control their, choroidal neovascularization. But when you package it into a sustained delivery system and then put that directly into the eye, you see no systemic toxicity, and the doses are, magnitudes of, orders of magnitude lower than what you see systemically with like oral or systemic dosing. And so that's actually, I think, maybe one of the bigger benefits. And so often when we're going into clinical trials, we're actually maybe not even trying to show superiority. We're actually trying to show non-inferiority with less dosing and then also less side effects, usually, as a first go. But yeah, that's a really good question also.
Thank you.
Thanks.
All right. Let's see. There we go. Okay. Good afternoon, everybody. So it's a real honor to be here and talk about epigenetics and targeting the epigenome. Here are my disclosures. I love the concept of this because many times through this journey, I ask the same question about what does this imply, and it's kind of a winding pathway, so I think hopefully it'll make sense. I just wanted to start with what is epigenetics, since we're all kind of coming from a different background here. So I think it's very clear to say that, genetics are genes and mutations in DNA, but epigenetics is essentially everything else. So starting with how the DNA is wrapped, it's wrapped in different conformations and then wrapped around histones, and then those are formed into nucleosomes. And I think the key points are that all of this is, number one, reversible and modifiable, and two, it's druggable. And the best analogy to this is, when you think about, this is my wife's desk, so you can see her cords are nicely tightly wrapped and in a great formation, and we're ready to go on vacation, and you get those little dongles that the cords are wrapped up with, and so good for travel. This is mine. And so I argue that this is ready to go. I know where everything is. It's readily usable. But you can't travel like this. And certainly it's got a lot going on. So moving back and forth between those two, that's really the study of what epigenetics is and how you could potentially modify that. So why do you care, and what is the potential? I think the key is that, number one, you can potentially unlock an untreatable cancer or change its state and change its response. The other thing is that we don't have drugs that target this now, so it's a new addition to our cytotherapeutics or immunologic therapeutics. And in bladder cancer, there may be reason to think that this is actually a precision target. Now, there's a number of these drugs already available, and people are studying them, and they do any kinds of function to affect histones. I'm going to put a plug in for our breakout session that hopefully everyone will attend tomorrow. A lot of seating. Come join us. 10 o'clock, we have a great core of folks coming who are our faculty. So please come join us at 10 o'clock, and again, we're going to be hitting this in greater detail. So our journey for this really began for bladder cancer, and we started with this because we were interested in mouse models. I did this in grad school and said, let's study mouse models when I started our lab over a decade ago. And at that time, the only thing that was really done was this carcinogen model that many folks here use. When you gave the animals carcinogen, they get bladder cancer just like patients do. Now, these look like bladder cancers that we would see in our patients, and they form tumors all over the whole bladder, and the histology looks the same. So we thought we should credential that and say, is this real? Do the mutations matter, or do they not matter? And interestingly, this is just as Seth was finishing the TCGA for bladder cancer, and at the same time, they found about 70% of patients with bladder cancers have mutations in chromatin regulatory genes. And that was very unique for a solid tumor. We found that the mice had about 90% of them had a mutation in the same genes. And this is a whole pathway called MLL3, and these actually sit on enhancers and regulate conformation. The mutation location is the same in mice as it is in humans, and again, this is showing where along the gene it's located, and again, these are very similar. So what does this mean? That was our first question. Again, our thought was that there's a balance. So if you have a mutation in these green set of genes, these are the MLL3 chromatin genes that open up chromatin and they're mutated, then that suggests on the other side there's an imbalance, and that the things that are closing the chromatin may be in sort of winning that battle. And what was regulating that is this enzyme called EZH2. It's part of this complex that causes chromatin to be compact. And so we said, well, let's look at that, and we see its expression. This gene called EZH2 goes up in bladder cancer. You can see higher brown staining there in high-grade tumors. When you give the mice carcinogen, the amount of this goes up, so it seems to correlate with cancer. So we thought, well, if we knock it out, what happens? And when we knock it out, we find that the tumors go away. This is actually a drug. This is the only drug that was made available at the time from a company called Epizyme. And we found that when you take a mouse, you give it a tumor, and you give it this EZH2 inhibitor drug, that the tumors actually start looking smaller. The target goes down after about 72 hours. And certainly that the amount of red, which is shown here, which is the number of advanced tumors, goes away. From an expression perspective, they go from being very basal, which is what this model makes, to be much more luminal, and that just sort of goes along with the tumors regressing. So again, going back to Max's question, if this is true, what does it imply? Well, it implies that there's something going on, and mechanistically we weren't really sure. I would have argued that the tumors are just getting smaller because they're not growing, but in fact, it actually has to do with the immune system. And we had no idea to predict this at the time. When we looked at the tumors, we found that they were just packed with immune cells. These look like lymph nodes. And sure enough, when you count the number of T cells, that's just full of T cells. And right around the same time, we were scooped by this group that found that this may be due to antigen presentation. But it was a little bit interesting when we looked at the expression in the mice, we found that it wasn't MHC1, which all nucleated cells make. It was actually MHC2, which is not really what we would anticipate. So MHC2s are only expressed on professional antigen presenting cells like macrophages and other APCs. And we actually found that when you give a bladder cancer cell this drug, it becomes a professional antigen presenting cell. And so you can see that those whisker plots, all these MHC2 genes go up with expression when you treat with an epigenetic inhibitor. And so we did this sort of kind of important experiment where we took a bladder, ground it up after treating the mice with the epigenetic inhibitor and co-cultured them with T cells, and found sure enough they do act like antigen presenting cells, and the tumor itself, or at least the bladder, becomes a professional antigen presenting cell, turning it against the tumor itself. So then what do we do with that? Well, you know, Maha Hussein is my partner and collaborator at Northwestern, and she said, you know, if there's a drug, we should do a trial. So this trial was, she was the PI of this trial, was sponsored by the NCI, and many people in this room were involved in this trial from the ETCTN. It was started, I think, around 2018. This was a two-arm study in patients with metastatic bladder cancer. The first half of patients were platinum refractory, the other half were platinum eligible. Both arms are closed. The arm B just closed, I think, last week. And if you look at the Kaplan-Meier curve here for this was reported at GU ASCO, it's not amazing, but if you look at the response data, it's actually not too bad. So we had 25% of patients that had a partial response, 3 had stabilized disease, and about a third of them had progression. This was all going on during the height of the pandemic. So, you know, there were some people, for example, that got, you know, only 1 dose and essentially succumbed from the coronavirus. So, but if you look at the median number of doses, it was 14 weeks of treatment, and the range was 3 to 107. So there were certainly some patients that had an ongoing response to this, and we're sort of interested in studying this. I think the key point here is that there's only one drug targeting this whole pathway. It'd be great if there were more drugs targeting this that we could look at. Right now the company, again, was bought by someone else, and the only drug is really available from the NCTCN. I don't know if this is the right stage to be treating. I think, for example, we're treating metastatic patients with highly refractory tumors. This is a pill that potentially stops working after 3 days. So if you think about moving it up to earlier stage bladder cancer, there may be a role. I also think that combinations and when you give it also is also important. So continuing with Max's question, if this is true, what else can we do? And I think, you know, again, this highlights Billy's work. So this is a great paper, one of my favorite papers in the last several years. He was looking at atinostat. This is an HDAC inhibitor. And really, in a very elegantly manner, he was able to show that you could change the neoantigens of a tumor and make it more susceptible to checkpoint therapy. Billy's got a trial for this. Again, come see him talk tomorrow at our breakout session. Our group is also interested in this other gene. It's, again, another epigenetic regulator called KDM5B, and we found that in tumors that don't respond to checkpoint therapy, this master regulator is overexpressed, and it represses a number of immune targets. And so, interestingly, when you look at the TCGA as well as our data, it's inversely correlated to immune infiltration, again suggesting it negatively regulates the immune response. If you treat with an inhibitor, you get activation of an immune response, as well as it looks just like FGF receptor 3 inhibitors with a lot of overlap in gene targets between them. And when you look at the molecular data, again, all of this is suggesting that if you treat with an epigenetic inhibitor, you can change the response of a tumor and change its phenotype. The problem, though, is that the structure's been solved in 2016, and the number of drugs is very few. So I think, you know, this is a major challenge. The drugs aren't as effective, and they have a lot of toxicity. So how do you get around that? Well, some ideas come from looking at the fly. So this is data that is from one of our collaborators looking at these enzymes because they go all the way back to yeast. You can find them in Drosophila. And so if you take a fly that has a regular full-length MLL complex, they're fine. If you delete it, the fly dies. But you can see here in the third bar that you can start truncating it. You can start breaking it up different pieces, and you don't need the enzyme. We think the enzyme is the important part in it. It probably isn't. You can get rid of it. You can get rid of all but 80% of it. And that seems to be the protein binding part where it binds another protein called UTX, and it does just fine. So maybe these epigenetic molecules really aren't enzymes. Maybe they're big protein structures, which really changes how we would target it. Instead of making enzymatic inhibitors, maybe we should be looking at something else. And that's where I think this field is really going to go, is that there's ways to target proteins. And that's sort of the next sort of stage of pharmacology is how do you target the protein itself? And there's different ways to target the protein for the proteasome so it can be degraded. And it really doesn't matter how you get there, but it's not just the enzyme, but the protein itself. And if you can target it for degradation, then you can really have maximal function. So in summary, to target the epigenome, I think we have a lot of good targets. They're great, but they could be better. And so more are needed. We need better therapies. I think we need more trials and more patients involved in these trials to really make those advances. Again, the enzymes are important, but they may not be the only part of them. And then I think degradation of the protein is going to be important in the future. Thank you.
All right, I'll get us started. So, that was a brilliant talk, and I feel like I learned a lot just listening. If we are going to look at so one of the things we're doing today is we're trying to look outside of bladder cancer and then bring it back in. Is there, I guess, a lessons learned that we know of epigenetic targeting, and is bladder cancer the right place to test these agents initially, or should we be going elsewhere and then coming back to bladder cancer?
Yeah, I think what I would say from that, Max, is that it's hard getting compound into patient, right? There's a lot of, if you look at the number of CTEP compounds, there's a couple of them that are potentially regulating DNA methylation, but really that barricade from compound, what we see in cells, potentially in mice, to getting into trials incredibly hard. There's a compound that we've kind of identified through a CRISPR screen, but making enough compound and getting that into people, that's a huge barrier. And those are companies that are made and bought and scientific careers lost from people trying to do that. So I think the science is really good. I think the translational part is hard, and that's where hopefully a session like this of getting people from different groups together to sort of say, how do we do that? To me, that seems to be that biggest gap is the science is beautiful, but then the hard part is getting those into patients that I think is the biggest issue.
Hey, Jess, thanks, great talk. Wondering your thoughts on trying to target switch SNF like remodelers, because those are frequently mutated in bladder cancer. We don't have time to talk about it now, but just the big picture thoughts on how targetable are they? Do you target them, or the immune sort of implications of their loss? What do you think?
Again, I think it comes down to enzymatic function and specificity, right? So if sweet SNF is essential for all of our cells, so how do you make it at that level where you can get either enzymatic targeting or maybe it's degradation that's the issue? I mean, that's one of the benefits of mucosal delivery. We've seen that with different devices we put in the bladder. So maybe that's the way that you go with that. So you need a drug and a delivery system. I haven't seen, in that same vein, it seems like there's other targets that seem to be altered in that setting that maybe we can go after as well. So I don't know, do you have any ideas?
No, I think that, the points you bring up about the essentiality makes sense, but maybe there's paralog dependence and things where, if you really have, say, ARID1A loss in a cell, that provides a vulnerability that you can target. Those drugs don't exist right now, or they're not very far advanced, but you maybe think about that sort of, dependency is the way to think about it, to target it. I don't know.
So, great.
Josh, congratulations on all your work. It's really fantastic. So you showed the TCGA work showing the mutations in KDM6A and other chromatin modifiers in bladder cancers. And as you know, there is work showing that these are also mutated in normal urothelium, and maybe those are some of the first lesions that result in this field defect. I'd love to hear your insights as to whether you think, you know, these mutations or even the functional consequences of these mutations are different early on in carcinogenesis or even pre-carcinogenesis compared to advanced bladder cancers.
Yeah, that's a great question. And so those were all in those Nature and Science papers a couple of years ago, where these little biopsies were done of people with normal bladders, and that was one of the earliest changes that happened. There's probably something there for the tumor initiation phase, right? We don't know exactly what that is because it seems intrinsic to bladder. I mean, I'll tell you that MLL3-4 loss is somehow associated with altered purine metabolism. Like, we're sort of seeing that as like a basic metabolism of DNA biology, of how that's occurring, and it's setting up some kind of fitness. But I agree, even in BBN, when we do this computational package to look at what are the earliest mutations, it's like P53 and the MLLs were like very early. So they're doing something to set up the urothelium, right? And there's probably a lot of biology there in pre-initiation phase. It's probably metabolism, but I don't know.
Beautiful talk. So you kind of showed us a pretty compelling heat map with the EZH2 inhibition, showing kind of luminalization of the BBN model. That might be important too as a therapeutic target, and I wonder if you have any ideas about how that works.
Well, again, I don't know if it's luminalization, David, or it's like you're getting rid of the tumor. Right, so that, you know, the urothelium is already very luminal, the tumors are very basal, so to luminalize them, the question is, are they still tumors? Probably not. They're mostly, you know, you're getting rid of the basal elements in there. So I don't know how much of that is epigenetic versus the tumors just going away. The part that we spent way too long trying to figure out is, are the tumors dying and then the immune system's eating them up, as opposed to running with the thought that we actually may be pulling the immune system in. And I mean, I wasn't smart enough to figure that out early. It took a long time to think about. It may actually be involved in recruiting the immune system. I don't think it's luminalization as much as like the tumors are just going away.
I'm going to go in a very different direction with maybe the last question, unless there are any others. So this idea of if it's true, what does it imply? I mean, you expressed it really well in that you showed us preclinical work into a phase 1 trial, which is, I think, what a lot of the trainees who are here, junior faculty who are here, all of us ascribe to do, but it's very, very difficult to do that full bench to bedside approach. I'm wondering what advice and what tips do you have to people who are sort of on that pathway and trying to get from point A to point B?
Yeah, I think the real thing the reason we ended up going to the NCI was we saw a result. We were pretty early to partner with a company and ask for drug to put in our system. And they said they don't have any money, but they gave it to the NCI, who said, yeah, let's do a trial. But again, so I think a lot of that was driven by just trying to show our data and try to, I think the point you made was like steal like an artist. So we try to pull other things into our field, and this was clearly a concept, a drug concept that was out there of epigenetics, and it made a lot of sense to us in bladder. So I just think we just kept trying, and eventually it worked. You know, we found a place that worked.
Thank you.
Thank you, Max and Shilpa, for the opportunity to come here and visit with all of you today. So we study pancreatic ductal adenocarcinoma, or PDAC, that's thought to be the model immunologically cold tumor that we actually think of being cold rather from the antigenic sense, as it has few mutations and thus fewer mutation-derived neoantigens, which we think of being as one of the reasons why PDACs in general have fewer T cells, which render them largely resistant to immune checkpoint inhibitors, and more broadly typify the nearly 80% of cancers or so that are similarly lowly mutated, cold, and thus immunotherapy resistant. But fascinatingly, although most are cold, a rare 9% of PDACs are in fact quite hot, with primary tumors enriched in about 12 times greater densities of activated CD8 T cells that we believe target mutation-derived neoantigens. And these tumors are also enriched in other immune cells that boost these antigen-specific CD8 T cell responses. So for the past several years, we've been studying these patients with really two questions in mind. How do they do this, and how can we replicate this? And today I'll share with you some of our work on selecting these immunogenic neoantigens and delivering these immunogenic neoantigens with RNA vaccines. Okay, so what do we know about neoantigens in human cancer? Well, we know that of all neoantigens in human cancer, most arise in non-synonymous mutations, of which around 10 to 30% are predicted to bind MHC class 1, mostly arising in single amino acid substitutions in passenger mutations. So to vaccinate against a hypothetical tumor that you see there, where a more immunogenic orange neoantigen spontaneously activates these orange T cells, yet also harbors less immunogenic variants, you can envision a variety of scenarios ranging from vaccinating against the yellow neoantigen doing nothing, the blue neoantigen and priming new blue neoantigen-specific T cells, the orange neoantigen and amplifying a pre-existing T cells, or perhaps in the ideal scenario, vaccinating against both blue and orange neoantigens to both prime new T cells and amplify pre-existing T cells. All of which requires a strategy to do two things. One, is select the immunogenic neoantigens and estimate dominance, for which we have been working on a neoantigen quality model. This model, we estimate a neoantigen to be more or less immunogenic based on features of non-selfness and selfness. And we estimate neoantigen non-selfness based on neopeptide sequence similarity to other known antigenic peptide sequences derived from infectious disease epitopes. And we estimate selfness based upon how the substituted amino acid differentially binds either the MHC or the TCR compared to the corresponding wild-type peptide sequence. This differential binding with respect to the MHC, we estimate there that you see as presentation as the ratio of the wild-type to mutant binding affinity to MHC class 1, and the differential reactivity with respect to a corresponding cognate TCR, we estimate based upon the relative ratios of the EC50s of this neopeptide sequence to a corresponding wild-type peptide sequence to a common TCR. This EC50 ratio that we estimate based upon the substituted amino acid position, size, hydrophobicity, and biochemical family. So in broad strokes, if the substituted amino acid looks too similar compared to the wild-type amino acid based upon how it binds either the MHC complex or recognizes a TCR, it is predicted to be less immunogenic, meaning it just looks too similar to the wild type to in fact generate an immune response. Now, with this neoantigen quality model, what we did was we looked in lowly mutated PDAC and a highly mutated melanoma and lung cancer, and what we found was that a neoantigen quality but not a quantity correlated with better prognosis, introducing the concept that tumor neoantigenicity could perhaps correlate with survival, and also introducing this concept that lowly mutated tumors such as PDAC could in fact harbor neoantigens that could be clinically targeted. Now, this was further supported by the observation that in these long-term survivors of PDAC, we were able to identify high-quality neoantigen-specific T-cell clones that were present in patients' archival primary tumors with identical T-cell clones persisting in the blood over a decade later, suggesting that these high-quality neoantigens could stimulate long-term immunological memory and perhaps were suitable as cancer vaccine antigens. Now, this is of course a phenomenological because we didn't have a control group in this observational study. So what we wanted to do was we wanted to ask, okay, well, if this is really true, namely these high-quality neoantigens are spontaneous targets in primary tumors of long-term survivors, then you would hypothesize that these high-quality neoantigens would be longitudinally edited over time, selectively in long-term, but not in short-term survivors, which would serve as a control group. So this is what we did here. We followed 9 primary tumors in long-term survivors out to 22 recurrent tumors over a 10-year time frame, and compared to a control group, what we found was that these primary tumors in long-term survivors, though they in fact had a longer time to evolve, they paradoxically evolved recurrent tumors with fewer mutations, which you see on the left of your screen, fewer immunogenic substitutions, fewer immunogenic tumors, as well as fewer immunogenic tumor clones. So based upon these observations, our working hypothesis was that these long-term survivors, they spontaneously generate these high-quality neoantigens that stimulate neoantigen-specific T cells that may delay PDAC recurrence. Now, interestingly, all PDACs, in fact, do harbor neoantigens, which raised the question, well, if neoantigen immunity could potentially be therapeutically boosted when not spontaneously immunogenic, as in the case of long-term survivors. So to therapeutically replicate what might be happening spontaneously in long-term survivors. Now, you might say, well, why individualized and why mRNA? Well, the reason for this was because these high-quality neoantigens in long-term survivors we found derived from tumor-specific passenger mutations, indicating the need for individualization, which we felt at the time the most flexible technology for rapid custom cancer vaccination in the clinic was to use mRNA. So this is back in 2017 now. We forged a collaboration between our group at MSK and Ira Melman at Genentech and Ugur Sahin at BioNTech to bring individualized mRNA neoantigen vaccines to treat pancreatic cancer, historically thought to be a cancer that did not contain mutation-derived neoantigens suited for such vaccines. And to test the hypothesis that in the adjuvant setting, individualized mRNA neoantigen vaccines could expand neoantigen-specific T cells to potentially delay PDAC recurrence, that we sought to test in a single-center investigator-initiated phase 1 trial where we targeted to accrue 20 surgically resectable PDAC patients on whom we performed surgery to remove their primary tumors in New York, and then within 72 hours we shipped their tumors to colleagues in Mainz, Germany, who then sequenced the tumors, identify the neoantigens, and custom manufacture individualized vaccines for every single patient. Now, while this is ongoing, back in New York, we treat patients with a single dose of atezolizumab at 6 weeks after surgery, and at 9 weeks after surgery, we benchmark patients to receive their first dose of their priming doses of their custom-made vaccines, followed by standard of care modified FOLFIRINOX and a vaccine booster. Now, we treated our first patient on this trial in December of 2019, and of course, shortly thereafter found ourselves conducting this complex clinical trial in the midst of a global pandemic, which posed several unique challenges, not to mention clinical and research shutdowns. There were global supply chain disruptions, which was quite critical for this clinical trial because this involved real-time cross-Atlantic transfer of patient material and drug, and of course, not to mention BioNTech now had the added slight distraction to make mRNA mRNA vaccines and save the world. But thankfully, despite all of these challenges, we were in fact able to accelerate the trial and last year complete this trial despite the pandemic a full year ahead of schedule. Now, though we targeted to accrue 20 patients, we ended up needing to treat only 19 patients with atezolizumab, of which 16 patients received vaccine. So overall, only 1 of 19 or 5% had vaccine non-manufacture. Okay, is rapid custom cancer vaccination feasible? Short answer is yes. You can see here our achieved time to atezolizumab was within 1 day, and more importantly, achieved time to vaccination within 3 days of benchmark times, despite us having to run this trial in the midst of a pandemic. So rapid custom cancer vaccination is feasible, fully integrated into a standard clinical workflow, and even after complex oncologic surgery as is required for PDAC. Okay, do individualized mRNA neoantigen vaccines stimulate T cells in pancreatic cancer, a lowly mutated cancer, historically thought to not contain such mutation-derived neoantigens for vaccines? So to answer these questions, we use 2 tests to monitor for vaccine-induced neoantigen-specific T cells, a primary test which measures ex vivo interferon gamma ELISpot responses in the peripheral blood, and a second, a confirmatory test to identify vaccine-induced neoantigen-specific T cell clones using TCR-V beta sequencing, a custom assay that we developed in our lab that we call CloneTrack. Both assays that preferentially detect high magnitude vaccine-induced T cell responses. Now, if you just look in the peripheral blood using standard flow cytometry, looking here at Ki67 expression in gray on CD8 T cells, PD-1 expression in red on CD8 T cells, and a LAG3 expression in blue on CD8 T cells, you can see after that first blue arrow a rapid proliferative burst with increased CD8 T cell expression of Ki67 that overlaps with upregulation of PD-1 and LAG3. You can then see that coming back down to baseline, and then after about 5 doses of the vaccine, which are indicated by those green triangles there, you see a similar proliferative burst with increased Ki67 expression on CD8 T cells, albeit to a slower magnitude, but that also overlaps with upregulation of a PD-1 and LAG3. We think the first peak is a PD-L1 response, and the second peak is a vaccine response. Now, to deconvolute the two, we obtain a pre- and post-vaccination blood for interferon gamma ELISpot that you see at the bottom of your screen. And in this patient who had 7 total targets in her vaccine, you can see there no responses to 6 of the targets, no pre-vaccination response to the 7th target, but a robust post-vaccination response that is maintained even after chemotherapy. Now, to identify the individual clones that are in fact mediating this response, what we do is we obtain all of the peripheral blood and we sequence and identify individual clones based upon their unique V-beta sequences. Now, if you just plot the rank frequencies of these clones in the peripheral blood, you get something that looks like this, which is not very informative at all. But what we do is we say, well, if you expect vaccines to induce clonal expansion in the peripheral blood, then you would expect that these clones to expand and contract similarly in the blood over time, allowing us to now cluster clones, not based upon their unique RNA expression profiles, but based upon their unique vaccine-induced clonal trajectories in the peripheral blood, as shown in this UMAP here, where every dot is an individual T cell clone, and its position on the UMAP, its unique vaccine-induced clonal trajectory in the blood. So you can see here that there's most of the clones are all clustering together, indicating that they have very similar trajectories. However, you have these clones over here up in the top left corner that look very different compared to all other clones, yet very similar to each other, suggesting they may in fact be vaccine-induced clones. That when you look more carefully, you can see that these clones are essentially undetectable at baseline baseline, and they increase about 10,000-fold with vaccination, reaching as high as 10% of all peripheral blood T cells. Now, interestingly, these clones are distinct from PD-L1 expanded clones, which are in fact down here, and this distinction becomes even more evident when patients get their vaccine booster, which is indicated by that small green triangle there, where you see expansion of vaccine expanded clones, but you do not see that for PD-L1 expanded clones. Now, if you ask, well, okay, what do we think these vaccines are doing? Are they priming new T cells, or are they amplifying pre-existing T cells? What you can see here is, though we do find PD-L1 expanded clones in the tumor, we do not find vaccine expanded clones in the tumor, suggesting that these vaccines are in fact priming new T cells to these antigens. But importantly, are these T cells in fact neoantigen specific? So to answer this question, what we do is we obtain the peripheral blood at the maximal time point of expansion, and then we re-stimulate the peripheral blood in vitro with the antigens that we include in patients' individualized vaccines. And then we sort and sequence the activated T cells in vitro, and we examine the overlap between the in vitro activated and the in vivo expanded clonal pool. And then for select clones, we use single cell sequencing and TCR cloning to confirm specificity. So I'm just show you two representative examples here, showing at the top here that in this patient we see 28 clones expanding in the peripheral blood with vaccines, and you can see here that of these 28 clones, 16 are in fact neoantigen specific that we confirm in vitro. And similarly, when you see even a smaller magnitude clonal expansion in the peripheral blood, as in the case here where you only have 9 clones expanding, you can see here that 3 of these clones are in fact neoantigen specific in vitro. So overall, we find that when we do see clonal expansion in the peripheral blood, that a large fraction of these clones in the blood are responding to the specific antigens in the vaccine. This is not, we believe, a non-specific response to the RNA itself. Okay, this very nicely correlates with the fact that our clone track induced clonal expansion that you see on the top correlates very nicely with measured interferon gamma ELISpot reactivity that you see there on the bottom. Okay, so these are the two tests, a primary test of interferon gamma ELISpot and confirmatory test of clone track, and we adjudicate a patient to be a individualized mRNA neoantigen vaccine responder if they have a positive primary test and a positive confirmatory test as adjudicated by teams that were blinded to both the clinical results as well as to the assay results. And with these criteria, what we find is that individualized mRNA neoantigen vaccines induce T cell responses in 50% of unselected PDAC patients. We find that these vaccine-induced T cell responses are quite substantial in magnitude, reaching as high as 10% of all peripheral blood T cells, and appear to be durable, lasting as high as 2.5% of all blood T cells, even 2 years later. Now, when we look at a median follow-up of 18 months that in fact extended beyond our pre-specified secondary endpoint of 18-month RFS, we find that vaccine responders have a median RFS that's not reached compared to non-responders who have a median RFS of 13.4 months. So suggesting perhaps that vaccines may delay a PDAC recurrence. Now, we think this could in fact be a true correlation between vaccine response and delayed recurrence, because we do not see similar correlations between other potential confounding variables, for example, a PD-L1 response and a delayed recurrence, nor do we see that these represent primary differences in patient selection, as we see equal representation of patients with lymph node status and resection margin status in both responders and non-responders, nor do we see differences in the difference of cycles of chemotherapy administration between the two groups, nor do we see differences in baseline immune infiltrates in these tumors. So the responders and non-responders have essentially the same number of global T cells, CD8 T cells as well as CD4 T cells to start with in their tumors. Okay, so you might say, well, okay, but maybe these non-responders just have bad immune systems as evidenced by their inability to generate an immune response to the experimental vaccine. So to answer this question, we asked, okay, well, can we measure immune responses in these non-responders to the experimental vaccine to a concurrently administered unrelated mRNA vaccine, which in this case was SARS-CoV-2 vaccination. And what we find here is that these non-responders, they mount equivalent humoral and cellular responses to a concurrently administered unrelated mRNA vaccine, suggesting that these patients do not just have a bad immune system, so these correlation between vaccine response and delayed recurrence is not confounded by differences in host immune fitness. Now, finally, though our trial was not in fact powered to detect differences in these metrics, looking for potential biomarkers of vaccine response, we found it interesting that even with this small sample size, that these vaccine responders tended to have more clonal tumors, and within these responders, if you look at neoantigen quality's ability to discriminate between immunogenic versus non-immunogenic antigens in the vaccines, the quality model was in fact able to discriminate between immunogenic versus non-immunogenic antigens here identified based upon ELISpot. So it's suggesting that there can be sequence-based features that can allow for rational antigen detection and incorporation in custom vaccine design going forward. Okay, now why are patients not responding? So interestingly, though, this first generation of RNA vaccination delivery mechanism is different from the COVID RNA vaccine delivery mechanism. Here we administer RNA vaccines intravenously for a maximal systemic lymphoid organ targeting, and it preferentially is enriched in the spleen. And in fact, in mice, a splenectomy abrogates the immune response by around 50%. So it was notable that for pancreatic cancer, as you may know, some of the surgeries involve splenectomy. So here you can see that there, although this did not achieve statistical significance, of course it was interesting that these non-responders were slightly enriched in patients who had their spleens removed. So this is our best working hypothesis going forward to why the immune response was only 50% in this trial. Okay, so in summary, we think mRNA neoantigen vaccines are an encouraging therapeutic signal in pancreatic cancer. We think that these vaccines are feasible in clinically relevant timeframes, fully integrated into a standard clinical workflow, and even after complex oncologic surgery. We think these vaccines are in fact quite immunogenic, substantially and durably expanding T cells in 50% of unselected PDAC patients, and that vaccine immunity correlates with a delayed recurrence, which we think is sufficient rationale for future broader testing in pancreatic cancer with a follow-up randomized phase 2 trial, which is now open here at MSK and accruing in this randomized phase 2 clinical trial. Essentially what we will be testing in the experimental arm is a modified version of the treatment schematic that we used in the phase 1 trial compared to a standard of care control arm. Now, just to end with, well, what can we really learn from this going forward, and how might we take neoantigen cancer vaccines more broadly to treat other cancer types? Well, we think this requires a series of 3 sequential steps. One is the need for a universal strategy to select the targets, for which we think the neoantigen quality model could provide one potential framework. And the second is to establish scientific rationale and clinical efficacy in a target disease for which we think pancreatic cancer, in fact, could be an ideal target disease to identify blueprints that could then be more broadly applied to other cold cancers and also potentially hot cancers, for example, bladder cancer. Okay, so with that, I'd like to thank all of the members in the team who did it. I'd like to make a special mention of Luis Rojas and Zach Sethna, who did all the work I showed you today, and also my longtime collaborators, Ben Greenbaum and Marta Wuksha. And thank you all for your time and attention. Happy to take questions.
Hi, Gary Steinberg from Rush. How long does it take you to make from actual pancreatectomy to you have a vaccine that you can inject into the patient? How long does that take?
So in this clinical trial, the benchmark time was 9 weeks, and our achieved time was 3 days of 9 weeks.
Okay, so in 3 days or 3 days less than 9 weeks, you said?
So the median achieved time was within 3 days of the 9 weeks.
Okay.
But this is our we would say first iteration, or in the early stages of learning on RNA vaccine design. And as we all know now, you can make RNA vaccines very fast. And we think this 9-week time frame is only the beginning, and certainly in future iterations. In fact, in our randomized clinical trial that I just quickly showed you, our estimated time from surgery to vaccine delivery is 6 weeks.
Because I'm working with a company, Epivax Therapeutics, and they use Janus Matrix as their neoepitope prediction model. And one of the things that's important, as you pointed out, but they really strive to eliminate all the tolerogenics, and so really just have an immunogenic vaccine. And we're shooting for 4 weeks in an mRNA vaccine, although I don't know if they'll ever have money for it, but we're shooting for 4 weeks because we're trying to design a neoadjuvant trial. And we think that neoadjuvant, starting in 4 weeks to start vaccinating, and then either concomitant or afterward using the checkpoint, because we think there's animal data to suggest that you're better off giving the vaccine prior to or concomitant to the checkpoint rather than the checkpoint first. Do you think that with Moderna's approach, you'll be able to reduce that down to even 4 weeks potentially, so you can use it in the neoadjuvant or metastatic setting?
Yeah, I think the ability to further optimize RNA manufacture and accelerate individualized vaccines in real time is certainly there. Theoretically, you can make vaccines in a weekend. So I don't think that the 9-week time frame that we did in this early learnings of clinical application of these drugs is a upper ceiling limit.
Do you think it's worthwhile to take something off the shelf and then add your personalized vaccine, or do you think that that's?
Yes, to answer the prior question on neoadjuvant vaccination, I think one at least our motivation behind doing an adjuvant vaccine study was really twofold. One was the observations that these immune responses spontaneously in these long-term survivors that we detected occurred in post-resected patients. So this was sort of in the adjuvant setting, if you will. And also the second is that vaccines historically have always been effective both in cancer as well as in infectious disease settings in the MRD setting, and not really in patients who have visible or certainly not advanced disease. So as we think that at least the first application as a monotherapy would likely be in patients with MRD. This is our thinking when we designed this, and this is also our current thinking as we move ahead with the phase 2 study.
Yeah, the nice thing about Bladder is that you can do a TURBT, get all the tissue you need for your vaccine, debulk, and so I think that as a model system in the bladder, if we could push forward in a neoadjuvant setting, I think that would be a nice thing to do.
Yeah, I think it remains it's an unknown question whether doing it when there is a visible tumor will in fact impair immune responses. The preclinical data actually suggest that with mRNA vaccination, the magnitude of the immune response sequentially decreases as the tumor gets larger and larger. So there do appear to be host factors that are important because these are in fact prodrugs which requires optimal host immune functioning to generate the response. This may not be ideal in patients with visible cancer, so this might be in fact a barrier for optimal magnitude responses in neoadjuvant patients.
Thank you so much, Petros Grivas, University of Aston, and Fred Hatch. Fantastic talk and really exciting, inspiring data. And obviously looking forward to the next step in the trial you have in Memorial. Picking back on the discussion that Gary started with the tissue and of course the time to generate the vaccine. I'm just trying to think about potential clinical trial designs, right, in bladder cancer arena. And if we think about an adjuvant trial, right, with MRD, right, as a concept that you just introduced, the question is, should we use TURBT tissue versus radical cystectomy tissue, and whether we think there is any evolution, right, in the clonal evolution of the new antigens, and does this matter? So what tissue should we use? And if we were to use archival TURBT tissue, would that be, and this is also a question for the, Josh, Max, and others, would that be a feasible thing to do? Because part of this tissue is being used for diagnosis staging. Can we get an IRB approval to use that tissue for research purposes? Is that feasible versus using the cystectomy tissue that, of course, if the timing is more than 6 weeks, will introduce some delays in the delivery of vaccine? So any insights in that regard about tissue of origin, clonal evolution, and feasibility of getting private tissue versus radical surgery tissue? Thank you, and fantastic work.
Thank you. In terms of neoantigen overlap between different tumors at different tissue sites, I shared these data briefly here that at least in pancreatic cancer, when you look at evolution of tumors from primary to metastasis, which are primarily in the lung, there is in select patients editing of targets, which means from a clinical application standpoint, if you have a patient with metastatic cancer, this raises the question, where do you do the biopsy from? Do you do it from the primary tumor? Do you do it from the metastasis? Or do you do it from both? And what do you do when patients have multiple sites? Then this might be because the antigens that might be immunogenic in each site may in fact not be overlapping. So this was part of the reason why we did it in the early stage resectable patients also, because there you have a single site, and this eliminates this question of clonal heterogeneity between different tissue sites. So I think this certainly can be answered, though, for example, in bladder cancer by retrospective analysis of these of exome sequencing from these two sites and just checking to see whether or not there's overlap or not. In terms of archival tissue, this does depend a little bit on the type of the computational pipelines required for RNA vaccine manufacturing. If RNA sequencing is used for RNA for neoantigen selection, then tissue quality becomes quite important, which requires an effort to minimize out-of-body time. So in this trial, we designed a whole series of staff to be able to ensure out-of-body time from out of the OR or out of patient to fixation within 15 minutes. So this would really optimize tissue quality so that it can allow for no dropouts because of inability to make the vaccines.
Thank you. This is Omar Al-Halabi from MD Anderson in Houston. Fantastic talk and fantastic paper in Nature. I have 3 questions, if I may. Number one, I don't know if with the numbers that you have, if you have data to look at clearance of ctDNA, in the responders to the vaccine, if that was able to clear whatever positive ctDNA they might have had. And second question, within the current assays to develop ctDNA, where it's basically tumor-informed type of ctDNA testing, can that algorithm talk to the sort of Moderna or the BioNTech vaccine development? So meaning the same time that you're customizing a ctDNA detection, customize the vaccine based on those, top header mutations that you're detecting. And then lastly, since you mentioned with the splenectomy, sort of abrogating the effect, do you foresee a B cell role rather than T cells? Because, traditionally diseases that are based on humoral immunity are the ones that are seen in patients with splenectomy, like meningococcal disease, etc.
So to answer the first question in terms of ctDNA, so pancreas cancer right now, based upon existing ctDNA detection strategies, in resectable patients, MRD, we detect around the fraction of patients that are in fact have detectable ctDNA ranges around 25% or so. So in this clinical trial, it was also the same. We did check it, didn't share the data today, but only around 25% of patients had detectable ctDNA in the periphery. So it's not, we didn't feel it was a strong enough, there was not enough sample size there for us to really be able to use that more broadly as a longitudinal marker for disease monitoring. In terms of your question of overlap between a liquid biopsy-based neoantigen selection versus tumor tissue-based neoantigen selection? I think that's a two-part answer. Number one, we do need to have a higher fraction of detectability in patients. So if only 25% have detectable ctDNA, then obviously this is a bit of a limitation in the ability, number of patients we would be able to make vaccines for. But also we would have to do overlap analysis to see whether or not the antigens that are found in the periphery include the immunogenic neoantigens. So this is work that is currently ongoing. In terms of the B cell question, the antigens that are included in these vaccines are primarily class 1 antigens. So, and for cancer, as you know, different from viral vaccines, the ideal targets that we would like to generate tend to be CD8 T cells. These are, we think, the primary cytotoxic cells which are essential for this. We have not excluded the possibility of there being CD4 T cells that are also expanded with the vaccines, but in this early analysis, based upon these assays that preferentially detect high magnitude responses, we see primarily CD8s.
Yep. The reason I asked about B cells, there has been some data with neoadjuvant bladder trials on tertiary lymphoid structures and the need for an orchestrated immune response with both components. So I don't know if you can see that with does these vaccines induce increased tertiary lymphoid structures,
Right. So in those scenarios, I believe those are studies where the tumor is in situ and you are either treating with some immune therapy pre-operatively and then examining for TLS formation in tumors. This is adjuvant, so tumors are removed. So at this point, there is in fact no tumor for us to generate a TLS in. So, but whether or not they generate B cell responses and humoral responses, I think is an interesting question, certainly not answered yet.
Thank you.
Can I just ask one question about the choice of the immune system? So I saw that you gave one drug, one dose of Atezo, which is kind of interesting. So number one, why a PD-1? Why one dose? I saw that in your newer schema, you're giving a bunch with the FOLFIRINOX, I think. I would think maybe a CTLA-4 would have more of a global modulatory effect. And then the second part relates to the spleen. And in bladder, we do a relatively reasonable lymph node dissection. But I always wondered, maybe we're removing the wrong lymph cells. Like maybe it's a better thing to leave those in since those have been the closest to the bladder, especially if we're pulling the immune system in. I mean, there's now some data that maybe removing more lymph nodes doesn't help us, that maybe that actually is worse. But is there any thought about that at all?
Sure. So the rationale for the sequencing of the drugs in the first trial, we gave, we intentionally designed it for sequential non-overlapping drug design, meaning PD-L1 first, vaccine second, chemotherapy third, so that we can deconvolute immune responses, specific contributions to every single drug. So by using TCR sequencing, we can find what are the clones that are responding to Atezo, what are the clones that are responding to vaccine, and what happens with respond to chemotherapy. So this was the rationale for non-overlapping. The rationale for combining was because was twofold here, because we anticipated or hypothesized that the vaccines would do one of two things. Either it would amplify pre-existing cells or induce de novo cells. If these are pre-existing cells, that presumably these would be inhibited by PD-1, PD-L1 pathway because these would be already pre-primed and present in the tumor. So by having PD-L1 pathway blockade on board, you already are disinhibiting the pathway and then further amplifying the cells with antigen delivery. So this was the rationale for combining it to really maximize clonal expansion. Why we would only give one dose, the reason for that was, the receptor occupancy for PD-L1 is extremely long, on the order of months. So we thought that even with a single dose, we were quite confident that there would still be receptor occupancy on board based upon prior work and knowledge on the half-life of this drug. So we felt that was sufficient evidence for us to think that there will be PD-L1 pathway blockade on board at the time we would initiate vaccination. In terms of lymph nodes, so the one interesting question from this is that, where do these clones come from? I briefly shared that doesn't seem like they're, at least in one patient, not coming from the tumor. We're doing a more extensive analysis to see whether or not these clones potentially are arising or pre-primed in tumor-draining lymph nodes or other lymph nodes or in the tumor. So I think this will hopefully help us understand what we should be doing with these tumor draining lymph nodes. For example, if these clones are coming from those lymph node basins, that perhaps this is a reason to maybe change our practice or not. But I think this is work that's ongoing. We hope to have some of that data out soon.
Phenomenal work. 2 questions. First question is, how does this compare to the previous trials in the pulse dendritic cells vaccination? I know they've been fairly promising in gastric and pancreatic cancer, and you're sort of cutting out that lead time for patients' own immune system to develop dendritic cells and expand their T cells. And the second question, in terms of the harvesting of the neoantigens, did you sample the tumor multiple times throughout multiple cores, or was it a one sample cut out? Because there's significant heterogeneity in terms of the expression profile.
Sure. In terms of a comparison of these results with other clinical trials of neoantigen vaccination, of course, as you know, it's a little bit challenging to do cross-trial comparison. The one finding that we were enthusiastic about was the magnitude of the response was very high, reaching as high as 10% maximal. This is over 1 billion T cells in patients targeting a single antigen to a target that was historically thought to not be immunogenic at all, which we were very enthusiastic about, to suggest that these are in fact bona fide targets in these cancers. These neoantigens exist in nearly all PDACs. I didn't share that data today, but we have done this analysis in prior work that nearly all PDACs have neoantigens. They are low in number, but they are there, and they can now potentially be therapeutically boosted with a vaccine. In terms of how many cores, so this was from a resected specimen. So this, what we do for tissue procurement is after we resect the specimen, the tissue gets grossed very rapidly within 15 minutes and gets fixed within 15 minutes of out-of-body time. And then after rapid path review by a dedicated pathologist, then we allocate tumor blocks which have maximal tumor content for vaccine manufacture. So it is sampling single area. We don't do multi-region sampling specifically for this. The multi-region question, in fact, a collaborator of ours, Christine Yacobuzzi-O'Donoghue at MSK, had prior done multi-region sampling on archival PDAC specimens, which had shown that at least in primary tumors, there was a fair amount of overlap between different sites within primary tumor, not from primary to met, but within one tumor. So this gave us a further ease that if we do a representative area, this would potentially be able to capture neoantigens or mutations would be found in other areas of the tumor as well.
So do you think these results are applicable to bladder and kidney, for example, where we see there's a significant difference in terms of the tumor composition?
In the primary tumor? So I think you rather than just looking at mutation number and overlap, it might be interesting to look at neoantigen heterogeneity specifically, because not all of the mutations are in fact neoantigenic. Right. The fraction that are neoantigenic or predicted is only around 10%. So it might be an interesting exercise to look to see whether the actual antigenic fraction is in fact different between different sites in the tumor. And if they are, perhaps is there some other way to be able to anatomically identify the sites so that you can capture them, for example, splitting the tumor in half and antigens from this and the antigens from that and creating a vaccine. These vaccines are polytope vaccines. So in this clinical trial, each patient gets 20 antigens in one vaccine in a single infusion. So there's no, and this is the 20 number is what is selected here, but there's no, in fact, upper limit cutoff necessarily based upon RNA design. So if you wanted to include more, for example, this certainly could be done.
So since there's no upper limit, you potentially could make one vaccine to cover multiple patients. Theoretically. So
not exactly. The reason for that is because these targets are there have been a lot of discussion, of course, in and I understand the motivation for this of why can't we just do off the shelf? Well, the reason for why we can't do off the shelf, at least this is perhaps a slightly biased opinion of mine, is our idea behind this, selecting the targets for vaccine has been to select the dominant endogenous target. In our studies in PDAC, we have found the dominant endogenous targets are patient-specific. They are not drivers. They're not RAS. Very small fraction of them are P53. So, of course, it would clinically make it much easier for us to have found that and said, okay, we could just do off the shelf because it's much easier. But we didn't find that, so we felt we had to do personalized because it just has to be personalized because those are the best antigens. What your question on could you do it more broadly, this raises a slightly different interesting question because even though it is personalized, there is only a limited space of substitutions within the substitution space. There's only 20 amino acids, so you could theoretically over time generate libraries of vaccines where, within a library you could contain potentially all potential or some fraction of the antigens that would be suitable for that particular disease type. So I think this would happen with time as we accumulate more evidence of what's the whole space of antigens that could be arising in a particular disease type, and then create sort of a vaccine library. That would be my thought on that.
Great. Thank you.
I have a couple of technical questions. How did you determine the dose in each vaccination, and how many vaccinations did you use? Did you use just one, or did you use it every week, or so? How many vaccinations should you use, and what's the timing, and what's the dose?
The dosing will likely be, I would anticipate, different based upon the particular RNA LNP formulation that would be used. Different companies have slightly different RNA LNP formulations. The one that we used was 25 micrograms of RNA LNP. This was based upon, there was prior to this, there was a phase 1 study, a safety and dose finding study in metastatic pan-cancer patients that did dose escalation and also had paired immune response. And based upon that, the 25 microgram dose was found to be the sufficient for high magnitude responses and with a favorable safety profile. How do we determine number of doses? If we I don't think I can go back there now, but if you looked at that on the clonal expansion graph, each triangle at the bottom was a particular vaccine dose. During the priming phase, they get 8 total doses, and you can actually see after only after around 5 doses of the vaccine do you actually see a huge spike in clonal expansion in the peripheral blood. So we do think at least 5 doses are required for clonal expansion, which is part of the reason why in the phase 2, in fact, the priming phase has 6 doses, because we think this is sufficient to be able to expand them.
Was that weekly or
This is weekly.
Weekly, so once a week for 6 weeks?
That's right.
Okay.
Yeah, in the first phase, of those 8 doses, the first 7 doses are weekly, then there's a 1-week break, and then you get the 8th dose.
Okay, thank you.
Thanks for the fascinating presentation, which, as you can imagine, is a little complex for the average layperson to understand. But I can tell you that those of us who are bladder cancer patients see the development of customized cancer vaccines as sort of the holy grail in treatment, kind of the penultimate of where immunotherapy might go. And we read in the lay press about other mRNA vaccines, particularly for melanomas, that are, I believe, in phase 2 trials. And I was just wondering how the work that you're doing compares with that. Is it vastly different, or is it kind of the same technology?
So the technology is similar. Both involve individualized RNA vaccines for cancer that are based upon individual genetic analysis of every single patient's cancer and real-time vaccine creation for every single patient. I think the difference between application of a vaccine in melanoma versus application of these vaccines in cancer such as pancreatic cancer the difference there is that melanoma, we already do know there are multiple ways to activate the immune system to treat melanoma. So certainly a RNA vaccine for melanoma is fantastic, and we would be very happy with that. But what is a high unmet need right now for the field of immuno-oncology is the 80% of cancers that the current first generation immune therapies do not work. And we think pancreatic cancer is a good example of this. So hopefully we can learn about how to apply RNA vaccines in such a immune therapy resistant cancer, and this can provide some information on how we would apply it to other cancer types in that whole 80% bucket.
And bladder cancer may be somewhere in the middle of that continuum?
That's right. That's right.
And the last question is, since this is so customized, I'm sure it's very expensive, so there's the financial part of it, but how about the scalability of it? And if we're not going to make an off-the-shelf vaccine, is this really scalable? You know, pancreatic cancer is a little bit less common than some of the other cancers. Do you see that becoming a problem?
So I think that's an important point that you bring up on, is such a customized vaccine really feasible for patients who are undergoing rapid or undergoing oncologic care, really need fast treatment? So these vaccines would need to be done very rapidly, and how broadly could you do it? So to answer the first question, was actually a primary reason for us to do the trial was to hopefully stimulate the community to say, yes, it is feasible. And this is also partly why we tried it in pancreatic cancer, because these are very big surgeries that we do for these patients. They require complex care after that. So if we can do it in pancreatic cancer, hopefully this will stimulate the field to say, well, it can be done in other cancer types as well. And with evolution of RNA manufacturing speed, which we now know, which we've always suspected, I think the vaccine manufacturing times are only going to get faster with experience. In terms of scale, I do think it is scalable. This particular clinical trial involved this strategy of cross-Atlantic transfer and so on, but certainly does not have to be this way. With local manufacturing, this could be solved.
Thank you again.
So I want to thank all of our speakers today. One of the main goals of the think tank is to be inspired, and I feel inspired. I hope all of you do, and I hope you'll take a lot of these lessons back to your peers at your home institution and the rest of the meeting. So I just want to thank all of you. That was wonderful.